So I'm somebody that grew up spending a lot of time outdoors, spent a lot of time interacting with nature and with biodiversity. And over the course of my life, I've just become increasingly fascinated with understanding what makes biodiversity and what makes species different from each other. And that fascination developed over such a long period of time is ultimately what brought me to the place where I'm at now, where I actually have an opportunity to really study and understand those aspects of biodiversity. I start my day at the University of British Columbia Herbarium, accessing the plant collection that exists there. It's one of the largest collections in British Columbia of plants, approximately 250,000 specimens collected from across the world. So these are housed in cabinets within the museum and stored into perpetuity so that researchers like myself can access them and use them for research. As a taxonomic researcher, I'm interested in assessing a variety of aspects of these plants, one of the most critical being the morphology, or the structure and shape of them. And so to do that, I need to have access to the actual real specimens that have been collected by botanists for over 100 years. So I'll bring those into the research room at the University of BC Herbarium, and I will have an opportunity to use the high-powered microscopes available to assess what are often very minute morphological details of the specimens. We use really nice dissecting microscopes to assess these little characters. The dissecting microscope allows me to observe things in 3 dimensions. So unlike a traditional slide microscope where you're looking at higher magnification, but you don't have that 3-dimensionality, this allows me to assess things like the structure of hairs, the shape of bracts and leaves in a real 3-dimensional kind of way. And so often these very tiny and very minute characters are the most important in separating very similar species. So, for example, the shape of the hairs and the stem might be really critical, as it was in some of the groups that I looked at for my thesis. And so having the opportunity to look at these specimens at such high levels of magnification and see such minute details, oftentimes I can be quite surprised by what I find. For example, the specimen that you're looking at here may well be an undescribed species from the Pacific Northwest. It has characters that are unlike any of the related species in the region. It grows in habitats that don't have any other species in this group. And so for a variety of reasons, it appears that this might be an undescribed species. And given the relatively few number of collections that we have of this species, indicates that it's likely quite rare, may well be endangered. And without being able to recognize it through taxonomy, we're not in a position to be able to conserve it or protect it or enable it to continue into the future. So one of the really important roles of the taxonomist is to help guide conservation efforts by recognizing these intrinsically valuable species. One of the major products that I develop is something called a monograph. And so what a monograph is imagine that you are a gardener and you're looking at a big garden with lots of different species of plants, and you want to know a lot of information about all of those species. So you may want to know when they grow, what kind of conditions they grow in, when they flower, how to distinguish different plants from other plants. And you want to have all of that information available sort of at your fingertips. And essentially that's what a monograph is. It's a taxonomist's version of that for all of the natural plants that occur within a particular group. I usually start by accessing the books available in the herbarium and compiling all of the information that is available in those books. However, now with the advent of the internet, we're able to do things as taxonomists that we would have thought inconceivable even 2 or 3 decades ago. So for example, I can go to the website JSTOR and I can have immediate access to high-resolution scans of the most important specimens pertaining to the species that I'm investigating. I can zoom in on them, I can assess them at microscopic levels of detail, and I can use those specimens to help inform my overall taxonomic system that I'm developing. In recent years, traditional taxonomy has generally been seen as a declining discipline, while molecular sequencing and genetic analyses have increasingly become the standard approach to answering so many biological questions. But I think that traditional taxonomy fulfills a pretty unique and valuable role in the biological sciences. It provides a framework for the interpretation of biodiversity and provides a common language that scientists and non-academics alike can discuss. That's why I think the best approach is for traditional taxonomy and modern techniques to work together, hand in hand, to more effectively answer the complex biological questions that we face.